Welcome to this next video in which we are discussing sarcoidosis. Okay, so we've now finished our discussion of the pathogenesis of tuberculosis, and we've seen that tuberculosis will result in this T helper 1 mediated adaptive immune response, which produces these granulomas around the Mycobacterium tuberculosis infection. And I hope that you now have some understanding of how these things form and what their purpose is from our discussion of tuberculosis. So we've seen that the purpose of these things is to wall off the Mycobacterium tuberculosis bacterial cells from the rest of the body. Okay? We can't eradicate the Mycobacterium tuberculosis cells completely in most cases, so we do the next best thing, which is we trap it in these little lesions and prevent it from spreading to other portions of the body. So I hope that that's given you an understanding of, at the very least, how granulomas form and what the purpose of a granuloma actually is. So now let's use our understanding to talk about sarcoidosis, and all of what we've talked about will transfer beautifully now to sarcoidosis. So, sarcoidosis, so let's just remind ourselves of sarcoidosis. In sarcoidosis we get granulomas forming in multiple areas of the body, but particularly in the lungs and mediastinal lymph nodes, which also happen to be areas where you're going to get granulomas forming in tuberculosis. In TB you'll particularly get granulomas forming in the lungs, but also in the mediastinal lymph nodes, because some of the bacterial cells will end up draining into the mediastinal lymph nodes, and there you'll get the granulomas set up around them. Okay, so sarcoidosis then, what do we think actually happens in sarcoidosis? Well, some agent is going to trigger the whole thing off, we believe. So there's some agent, and we will talk in a moment about different theories of what this agent actually is. So this is most likely a pathogen of some sort, and we'll discuss options, theories of what the pathogen actually is in just a moment. But at the moment we'll just refer to it as an agent. Some agent is going to come into the body. You're going to be exposed to some agent. So remember, a genetically susceptible host, that's the key term. Someone who is actually capable of getting sarcoidosis is now going to be exposed to their susceptible agent here. And most likely the way in which they're actually going to be exposed to it is inhalation. That would explain why the lungs and mediastinal lymph node are so often involved. But in those cases where you don't have pulmonary involvement and maybe you've just got cutaneous involvement, then maybe the exposure could be through the skin, but mostly it's probably going to be through inhalation. But you're going to be exposed in some way, so actually I'll call this next part of the flow diagram here exposure, okay, which will mostly be inhalation. So we'll draw a nice flow diagram for this. So this is the next box in our flow diagram, and I'll draw a little picture for this to go with it. So you'll inhale it down your trachea into your lungs here. So here's your right lung again, and here is your left lung. Okay, so inhalation will occur of the agent that you're susceptible to. And now, what's going to be initiated in response to this agent? Well, exactly what we've just gone through with Mycobacterium tuberculosis. When the agent is inhaled in, first thing that's going to happen is the alveolar macrophages and the alveolar dendritic cells are going to recognize its presence, most likely with pathogen-associated molecular patterns binding to pattern recognition receptors on their surface. The alveolar macrophages and the alveolar dendritic cells will then phagocytose the agent, whatever it is, and they'll also trigger a inflammatory response by releasing interleukin-1 and tumor necrosis factor alpha. So I'll make that my next box. So you're going to get inflammation occurring. Okay, so the acute inflammatory response will be initiated because of tumor necrosis factor alpha and interleukin-1. So you now see how studying TB has given us all the tools for understanding what's going to happen here. So we're going to get inflammation in response to the agent. We can't say as to whether the agent is actually going to resist phagocytosis in the way that Mycobacterium tuberculosis is going to resist phagocytosis, but potentially that is an option. We'll come on to that when we discuss the actual theories of what this agent is, but certainly it will initiate inflammation. And then what's going to happen is we must get the same sort of adaptive immune response initiated against this agent. We must get a T helper 1 mediated adaptive immune response. Now, why can I say that has to happen? Well, because the granulomas of sarcoidosis contain T helper 1 cells. They have to have come from somewhere. So we must get a T helper 1 mediated adaptive immune response initiated against our agent. So reminder now of what this will involve, and we've discussed this all in tuberculosis. So this will involve an antigen presenting cell, either an alveolar dendritic cell or an alveolar macrophage, phagocytosing the agent, breaking down the antigens of the agent into loads of little fragments, presenting those fragments on MHC class II molecules, and then going to the mediastinal lymph nodes to present it to T cells in the mediastinal lymph node, activating CD4 positive T cells with complementary T cell receptors to one of the MHC peptide fragment complexes, and those CD4 positive T cells must then become T helper 1 type cells, and we can't give the detail that we gave in the case of Mycobacterium tuberculosis. So remember, in the case of Mycobacterium tuberculosis, I told you exactly where the interleukin 12, which drove the effector helper T cells to the T helper 1 phenotype, came from. I said that the cells which are actually dealing with the Mycobacterium tuberculosis at the site of inflammation or the site of infection, they will have the toll-like receptor 2 pattern recognition receptor activated by the lipomannan of Mycobacterium tuberculosis, and this will cause them to release interleukin 12, which will then cause the production of T helper 1 cells. We can't say that sort of detail in this case, but we know again that something along those lines must happen. We must get interleukin 12 driving a T helper 1 mediated CD4 positive T cell response. So we're going to get T helper 1 cells produced against the antigens of our agent. These will go into the bloodstream, they'll come to the site of infection, and then what they're going to do is they're going to result in the formation of a granuloma around the offending agent. Okay, so what we now get is the formation of a granuloma. Now, the other thing that we can say is going to occur is that the granuloma in this case, in the case of sarcoidosis, it's not going to actually contain whatever the agent is. So remember, in Mycobacterium tuberculosis infection, provided that your adaptive immune response was strong enough, then these granulomas were capable of containing the infection. However, in the case of sarcoidosis, this doesn't seem to happen. The formation of the granulomas doesn't successfully contain the bacterial infection. It's not necessarily a bacterium, although the major two theories are that the agent is a bacterium. But the agent, they're not going to contain the agent, and the agent is actually going to disseminate. So, unfortunately, the granulomas don't seem to contain it, and you get dissemination. Okay? And then it can go elsewhere in the body. So, for instance, from the lungs, so I'll actually just draw a little picture of a granuloma forming here. So here's a lung with a granuloma forming there, and now it's not managed to contain it, so now the agent disseminates and it can go to other tissues, for instance the skin, where it can lead to granulomas forming there as well. So you get the disseminated granulomas forming, which is characteristic of sarcoidosis. So here is my picture of the skin with the granuloma having formed in the dermis of the skin, so you get dissemination. Okay, and that then is sarcoidosis, when we've got these disseminated granulomas all over the place. So the granulomatous reaction doesn't seem to be capable of containing the agent in the case of sarcoidosis. Okay, so that's our basic understanding of sarcoidosis, and you can see now why I took the time to go through what happens in the tuberculosis pathogenesis, because a lot of what we've been through, for instance the T-cell activation pathway, all of that is exactly the same. It must be exactly the same in this pathogenesis of sarcoidosis. So we can gain a lot of understanding just by looking at the pathogenesis and the immune response to tuberculosis. So, what I want to now discuss is theories for what the agent is, but before we just discuss that, let me say that we know something about the granulomas of sarcoidosis. I just want to discuss the granulomas of sarcoidosis in a bit more detail. So remember, the granulomas of sarcoidosis, they look very similar to the granulomas of tuberculosis, incredibly similar actually to the granulomas of tuberculosis. The difference is we call them non-caseating granulomas because generally there isn't the region of caseating necrosis in the centre of the granuloma that there is in the tuberculosis granulomas. However, as I said right at the beginning of the video, that is not a hard and fast rule. Often in sarcoid granulomas you do find a region of caseous necrosis right at the centre of the granuloma. Okay, so it's not a hard rule saying that there's no caseous necrosis in the granulomas of sarcoidosis. What I want to discuss, however, isn't to do with that, it's to do with the T helper 1 cells that you find in a sarcoidosis granuloma. So if you look at a granuloma of someone with sarcoidosis and you look at the T helper 1 cells around the periphery of it, you find that these T helper 1 cells are oligoclonal. Okay, so I'll put that fancy word. So we find that the T helper 1 cells of a sarcoid granuloma, and by the way, when I say a sarcoid granuloma, sarcoid is just short for sarcoidosis. It's a shortened version of the word that we use a lot. Okay, so when you look at a sarcoid granuloma, you find that the T helper 1 cells are oligoclonal. Now, what does that mean? So we describe them as oligoclonal T helper 1 cells. Well, oligo means some. Okay? Clonal refers to the clone of the T helper 1 cells, the type of T cell receptor on the surface of those T helper 1 cells. So what this is actually talking about is the fact that the T helper 1 cells in the periphery of a sarcoid granuloma actually have different clones. They have different T cell receptors. So you find a mixture of different T cell clones in the periphery of a sarcoid granuloma. You don't just find one clone of T helper 1 cells. Of course, there'll be loads of copies of the same clone of T cell in the periphery of a sarcoid granuloma, but some of the different T cells that you look at will have different T cell receptors, i.e. there are multiple different clones of T helper 1 cells in the periphery of the sarcoid granuloma. That suggests that it is a pathogen that we are dealing with here. It suggests that the agent that we are dealing with has multiple different antigens that have been cut up to give fragments that we have had the T helper 1 mediated adaptive immune response initiated against. Of course, they could all be from the same antigen potentially, i.e. they could all just be T helper 1 cells directed against different fragments of the same antigen, but it suggests that there is a pathogen with a collection of antigens that have had T helper 1 cells produced against them. Okay, right. So now what I'm going to do is talk about the different theories of what this agent is. Now, this is the bit where I get very, very controversial. So what I'm about to say is still very, very controversial. This bit is more or less agreed upon. Everyone can agree on this pretty much. What people cannot agree on is what this agent actually is that is causing the formation of the granulomas. Oh, the other thing that I should stress here is that you're only going to get this T helper 1 mediated adaptive immune response in response to the agent and the formation of these granulomas which disseminate if you are genetically susceptible. And actually, I shouldn't say that you're only going to get the T helper 1 mediated adaptive immune response. You're only going to get sarcoidosis in response to this agent if you are genetically susceptible. Now, that might mean that only in certain people, the genetically susceptible people, does this agent initiate the T helper 1 mediated adaptive immune response, i.e. in normal people, potentially, you don't actually need this. This doesn't occur and you don't get granulomas forming full stop. Alternatively, it might mean that you do get granulomas forming in normal people, but they just don't disseminate maybe, and therefore you don't get full-on sarcoidosis. So there could be lots of different reasons that this agent will only cause the disseminating granulomatous inflammation in people who are genetically susceptible. But remember that the agent only leads to sarcoidosis in certain people, so you must be genetically susceptible. Okay, so now what I'm going to do is discuss the two major theories of what this agent is and how the pathogenesis of sarcoidosis occurs then. And these two major theories are the Mycobacterial theory, which says that actually it's just part of tuberculosis, and then the Propionibacterium Acnes theory. Now there are loads of other theories of sarcoidosis. So for instance, there's a rickettsial theory of sarcoidosis. There are theories about how it's a pneumoconiacy, where you've inhaled some non-organic matter potentially that is causing the formation of the granulomas, and there are loads of other theories. So for instance, theories that it might be a prion infection, or theories that it might have an autoimmune origin. But the major two theories are the Mycobacterial theory and the Propionibacterial Acnes theory, and those are the two theories that I'm going to discuss. So let me just get another sheet of paper and we'll do this. So, the Mycobacterial theory of sarcoidosis then. So, when we've studied the pathogenesis of tuberculosis, I showed you a spectrum of different responses that you can get, or rather than responses, maybe I should call it outcomes that can occur because of being infected by Mycobacterium tuberculosis for the first time. So let's just go over what those different outcomes were. So, if you remember, when you initiated the T helper 1 adaptive immune response to Mycobacterium tuberculosis, the possible outcomes were that you could have complete eradication of the bacterium. Now that was quite a rare outcome, but it can occur. The most common outcome was that you would have latent tuberculosis. So firstly, let me just write on eradication. So if you like, that's at the best end of the spectrum. So over here, this can be the greatest end of the spectrum. So a complete eradication of the bacterium is one possible outcome from a primary TB infection. And remember, a primary TB infection is one of those infections where you have been exposed to the bacterium for the first time. So you're having to initiate the adaptive immune response from scratch. So complete eradication was one outcome. Then the next level of outcome up from that is latent tuberculosis infection, and this is where the adaptive immune response gets the infection under control. It gets it contained within granulomas. It stops it from spreading elsewhere, but it does not completely eradicate the bacterium from the body. Okay, so that's the latent tuberculosis infection. That's not quite as good an outcome as complete eradication, but it's not too bad. And then after that, right up here, we had the outcome which was not good at all. So this was what would happen in people who were immunosuppressed. You'd get progressive pulmonary involvement, so progressive pulmonary TB, which remember is an expanding granuloma inside the lung which will just gradually eat away the lung tissue. And you can also get it disseminating all over the body, and you'll get granulomas forming all over the body, so miliary tuberculosis. And that will almost certainly result in death if untreated. So that's the very bad outcome from a primary TB exposure. Okay, so the Mycobacterial theory of sarcoidosis is that sarcoidosis is just another outcome of a primary TB exposure. It's just part of this spectrum of outcomes from a primary exposure to TB, and it would be positioned here. So you'd put sarcoidosis here. And the idea is that in sarcoidosis, you get exposed to Mycobacterium tuberculosis for the first time. So this is the dreaded agent in the Mycobacterial theory. So the agent in the Mycobacterial theory would be Mycobacterium tuberculosis. You get exposed to it by inhalation Okay, you'll get inflammation as we've described, you'll get a T helper 1 mediated adaptive immune response, you get granulomas forming, and unlike in the latent tuberculosis outcome, unfortunately the granulomas can't quite contain the bacterium. Occasionally they leak. They're not quite good enough. They're not as bad as this. In this they're awful, and they gradually expand and expand and expand and certainly leak all over the place. Here though, they'd rarely leak. Occasionally they'd leak, and then you'd get Mycobacterium tuberculosis going elsewhere in the body, and you get another granuloma forming elsewhere in the body. So the idea is that the granulomas aren't awful here, but they're not quite as good as in latent tuberculosis. So there is some problem with the adaptive immune response against Mycobacterium tuberculosis. That is the Microbacterial theory of sarcoidosis, that because of your genetics, you aren't capable of fighting Mycobacterium tuberculosis quite well enough, and therefore you don't get latent tuberculosis, you instead get sarcoidosis. Okay, so let me discuss the evidence in favour of this and the evidence against this. So the great piece of evidence against this is quite simple and quite blunt, and the great piece of evidence against it is that if you take a sarcoid granuloma, you cut it up, you stain it with the Zeal-Nielsen stain, which remember stains Mycobacterium tuberculosis. What do you think the answer is? Well, the answer is you do not find any Mycobacterium tuberculosis. So you do not find Mycobacterium tuberculosis cells inside the granulomas of sarcoidosis. Whereas, if you were to look at a granuloma of miliary tuberculosis, for instance, or one of the massive granulomas in the lung in one of these outcomes of tuberculosis, you would absolutely find loads of the bacterial cell inside the granuloma. So, how does this theory cope with that? Surely that says, no, this is wrong. If there's no Mycobacterium tuberculosis in the granulomas, then Mycobacterium tuberculosis surely is not the agent that is causing the granulomas in sarcoidosis. Well, here's the reply of the Mycobacterial theory to this, the people who are in favour of this theory. You see, the whole idea is that sarcoidosis is a better adaptive immune response to TB than this. This is where the adaptive immune response has completely failed. The idea in sarcoidosis is that it's not actually too bad, it's just not good enough. So the idea then is that in these sarcoid granulomas, the adaptive immune response nearly controls the Mycobacterium tuberculosis. It gets the population to such a low level that it's below the detection threshold. So the idea is that the amount of Mycobacterium tuberculosis cells in the sarcoid granulomas are actually below the detection threshold, and therefore that this is actually in keeping with the theory that sarcoidosis is within the spectrum of outcomes of primary exposure to Mycobacterium tuberculosis. Okay, now, why does that sort of make sense? Well, you see, the thing is, if you were to look at one of the lesions of latent tuberculosis, so if you were to take a granuloma of latent tuberculosis from the lung, you would have an incredibly difficult time finding Mycobacterium tuberculosis inside that as well. Okay, there are some reports, but very few reports of actually being able to find Mycobacterium tuberculosis in latent tuberculosis lesions. It's one of the reasons that latent TB is so difficult to actually study, because it's so difficult to find. So it is really, really difficult to find Mycobacterium tuberculosis cells in the lesions of latent TB. So that's why this kind of makes sense. So here, where you've got the Mycobacterium tuberculosis under very good control, it's at such a low level that you can't really find it by the staining process. And I'll just put that this is by staining. So no Mycobacterium tuberculosis when you stain with the Zeal-Nielsen stain. Okay? You can also try and actually culture the Mycobacterium tuberculosis. So you can take the tissue and try and grow the Mycobacterium tuberculosis from that tissue on an agar jelly. So you can just take the tissue from a granuloma, mush it up a little bit, and then spread it out on an agar jelly. And the idea is that if it contains Mycobacterium tuberculosis, then you'll get the bacterial cell growing on the agar plate and you'll be able to culture it. That also fails in the case of sarcoidosis. You can't culture the bacterial cells from samples of sarcoid granulomas. So it's very, very difficult to find Mycobacterium tuberculosis cells in sarcoid granulomas. But that kind of fits because in the latent TB granulomas, it's also very, very difficult to find Mycobacterium tuberculosis cells in those granulomas because they're at such a low level. So they can't really be found with Zeal-Nielsen staining or with culturing mechanisms either. So the idea then is that in sarcoidosis, the granulomas that you've got are very good. They do keep the Mycobacterium tuberculosis at a very low level, below the detection threshold for these methods. However, they're not quite good enough, and the Mycobacterium tuberculosis does spread from the granulomas and then disseminate to other sites, so you do get the spread of granulomas around the body. Okay, so that's how the people in favour of this theory account for the lack of Mycobacterium tuberculosis in the granulomas of sarcoidosis. Now, let me give you some other sort of arguments in favour of the Mycobacterial theory of sarcoidosis. The number one argument in favour of this is quite simply the histological similarities between the granulomas that form in sarcoidosis and the granulomas that form in Mycobacterium tuberculosis infection. So histosimilarities, as I've written it there. So that's the greatest argument, that the granulomas that form in an active TB infection here look so similar to the granulomas that form in sarcoidosis. I've said that the granulomas in TB infection are caseating, whereas those ones in sarcoidosis are non-caseating. However, as I said, in sarcoidosis you can often find granulomas that are a little bit caseating, so that distinction isn't completely hard. Okay, the other great argument that people have now in favour of the Mycobacterial theory is that we now have more sensitive methods. It's now the era of genomics and proteomics. Okay, molecular biology has taken off in the past few decades, and we now have more sensitive ways of detecting the presence of Mycobacterium tuberculosis in the granulomas of sarcoidosis than just staining with the Zeal-Nielsen stain and trying to culture the bacterial cells from the sarcoid granuloma tissues. What we can now do is we can look for DNA of Mycobacterium tuberculosis cells in the granulomas of sarcoidosis using the polymerase chain reaction. We can also look for proteins of Mycobacterium tuberculosis in the granulomas of sarcoidosis. And when you do this, you can actually find it quite often. Okay, so you can actually find DNA and proteins from Mycobacterium tuberculosis in the granulomas. However, this is still very, very controversial. You see, loads of people have done these same experiments. There are loads of different groups around the world that have done the same experiments where they've looked for the DNA of Mycobacterium tuberculosis in granulomas from people with sarcoidosis, and they've also looked for proteins of Mycobacterium tuberculosis in the granulomas from sarcoidosis. And different groups have got different results. Some groups get that every single granuloma that they've taken from people with sarcoidosis has DNA from Mycobacterium tuberculosis in, whereas other groups just can't find it at all. So it's still very, very controversial. It's not clear at all who is right and who is wrong. So some people have found DNA and proteins of Mycobacterium tuberculosis using the modern techniques of molecular biology in the granulomas of sarcoidosis, whereas others have failed to find it. So that's still got a question mark by it. We don't know. However, that's one of the things that people in favour of the Mycobacterial theory always quote. They quote the studies that do find the DNA and proteins in the granulomas of sarcoidosis. So of course this would demonstrate potentially Mycobacterium tuberculosis is present in the granulomas of sarcoidosis at very low levels, below the detection threshold for these older techniques, but within the detection threshold for these newer techniques. Okay, so to summarise overall then the Mycobacterial theory of sarcoidosis, The Mycobacterial theory says that sarcoidosis is caused by Mycobacterium tuberculosis. In certain people who are genetically susceptible to Mycobacterium tuberculosis, these are the people who are susceptible to sarcoidosis as far as this theory is concerned, when they are first exposed to TB, okay, so when they get their primary exposure to TB, unfortunately for some reason, for some genetic reason, their adaptive immune response is not good enough to just give them a latent TB infection. Instead, what happens is they get the granulomas disseminating around the body. So you do get granulomas forming in the lung and the mediastinal lymph nodes around where the Mycobacterium tuberculosis was originally positioned, but they don't manage to completely contain it. They keep the population very, very low, so they're not terrible. They do keep the population very, very low, but they don't manage to completely contain it. And it does end up getting out of the granulomas, disseminating to other sites in the body, and wherever it goes, you'll get granulomas being set up, and that leads to sarcoidosis is the idea. And the theory would then say that the reason potentially that people get remission from sarcoidosis is that for some reason that we don't know, the body somehow suddenly manages to actually control the Mycobacterium tuberculosis better, and in all of the granulomas around the body that you now have where the Mycobacterium tuberculosis is present, they become latent granulomas where they have it completely under control. So the idea is that the reason that you can get remission from sarcoidosis is that it turns into a latent tuberculosis infection. So this is the explanation for remission in the case of the mycobacterial theory. Okay, right. So now what I want to turn my attention onto is the other major theory of the etiology of sarcoidosis, which is the Propionibacterium Acnes theory. So, let's now discuss the Propionibacterium Acnes theory of sarcoidosis. So firstly, let's just get over the name of this new bacterium. So, Propionibacterium Acnes is the star of this next theory that we're going to discuss of sarcoidosis. So, Propionibacterium Acnes, if you have heard of it before, it will probably be with regards to acne. It is believed to be one of the major causative agents of acne. So it's a commensal on the surface of the skin normally. However, in the teenage years where sebaceous output increases hugely, it can actually become disease-causing. It can infect the sebaceous glands and can then lead to inflammation there, which leads to acne. So, Propionibacterium acnes. So the first thing I want to do is actually study this bacterium in its own right, like we did for Mycobacterium tuberculosis initially. So P. acnes is the short name for it, so we shorten Propionibacterium down to P. Now, firstly, what shape is it? So it is a bacillus, just like Mycobacterium tuberculosis, so it's a rod-shaped bacterium. It is also non-motile like Mycobacterium tuberculosis, so it doesn't have flagella. It also has a single circular chromosome as its main genomic DNA. The difference between it and Mycobacterium tuberculosis is that, unlike Mycobacterium tuberculosis, which, if you remember, was neither gram-positive nor gram-negative, Propionibacterium acnes is actually gram-positive. So it has a simple peptidoglycan cell wall surrounding the cell membrane, like so. So it is a gram-positive bacterial species. So I'll just put this down here, so gram-positive. So here's my basic picture then of the bacterium Propionibacterium acnes. Now, as I've said, it's a commensal on the surface of the skin, and except in the teenage years where it can cause acne, it usually doesn't cause any problem having it on the surface of the epidermis. So just drawing a basic picture here. Here is the skin once again, the epidermis there, here's the dermis. So on the surface of the epidermis you have a huge number of different bacterial species that live there without causing a problem, and they're actually extremely important for preventing bad bacterial species from growing on the surface of the skin. Having a lot of bacterial species bacterial cells on the surface of the epidermis that aren't causing any problem means that there's a huge amount of competition for any other bacterial cell that wants to come and live on the surface of the skin. And this is a huge part of preventing bad bacterial species from growing on the surface of the epidermis. Okay, so that blue line is just meant to represent some Propionibacterium acnes cells on the surface of the epidermis. Okay, it's also a major commensal on the lining of the gastrointestinal tract in certain portions of the gastrointestinal tract. Okay, so it's a commensal on certain epithelia of the body. So, the Propionibacterium acnes theory of sarcoidosis. Before we can discuss actually how this is going to lead to sarcoidosis, we need to discuss the first part of this theory. And the first part of this theory is that actually, in a huge number of people in the human population, Propionibacterium acnes produces an endogenous infection of the lungs and mediastinal lymph nodes. And I must say at this point, this is the part where we go on to the theory. This is controversial whether this is true or not, but there is a lot of research, particularly in Japan, that backs up this theory. Okay, but this is controversial. All of this bit that we've discussed so far was not controversial. This is the structure of P. acnes, and it is a commensal on the surface of the skin and the gastrointestinal tract. Now we go on to the theory. So the theory is that this bacterial species actually causes something known as an endogenous infection in the lungs and the mediastinal lymph nodes. So the first thing that I need to explain to you is what is actually meant by an endogenous infection. Okay, so the idea then is that a lot of people around the world are going to end up inhaling Propionibacterium acnes bacterial cells into their lungs. And what will happen when the Propionibacterium acnes goes into the lungs? Well, before this theory came about, the answer would have been, well, it will get phagocytosed by alveolar macrophages and alveolar dendritic cells, and it will get eradicated by those phagocytes, and it will not be in the lungs anymore. However, what the Propionibacterium acnes theory states is that actually often the Propionibacterium acnes can go into a phenotypic state where it is latent, like the latent Mycobacterium tuberculosis cells that we've discussed, and is resistant to being destroyed by the killing mechanisms of the alveolar macrophages. So a lot of the Propionibacterium acnes cells that get inhaled will be destroyed by the alveolar macrophages, but some go into a latent state where their protection against the mechanisms is at a very high level, but of course the cost of this is that they're not then dividing very rapidly at all. So this is what it's going to look like. So here is an alveolar macrophage with the Propionibacterium acnes inside a phagosome. Okay, so the Propionibacterium acnes is there in blue at the centre. Okay, so the idea then is that some of the Propionibacterium acnes cells that get phagocytosed into into a phagosome like so, can go into a latent state where their protective mechanisms against phagocytosis are at the highest they can be. So they activate loads of protective mechanisms to stop themselves being destroyed, like we've discussed with Mycobacterium tuberculosis. But the cost of this is then the division has to be very low. Okay, so this is part of the Propionibacterial acnes theory of sarcoidosis, that in a lot of people you get the Propionibacterium acnes being inhaled, and some of these Propionibacterium acnes cells do not actually get destroyed by the alveolar macrophages and alveolar dendritic cells, and instead go into a latent state where they can survive inside the phagosomes of these phagocytes. Okay, so what happens now then? So the idea is that a lot of people do actually have alveolar macrophages inside their lungs and also mediastinal lymph nodes. So you have to understand that the lungs are drained. The lymphatic drainage of the lungs goes to the mediastinal lymph nodes. So these alveolar macrophages often do go to the mediastinal lymph nodes, and whoops, I've missed off the cardiac notch of the left lung here. Okay, so here is my basic picture of the lungs. Here are the mediastinal lymph nodes, shown here in green, like so. So we know that the lymphatic drainage from the lungs goes to the mediastinal lymph nodes. So if we've got alveolar macrophages in the lungs that have P. acnes bacterial cells latently sitting inside their phagosomes, then some of these will end up going to the mediastinal lymph nodes and sitting there, and therefore you will get these macrophages in the mediastinal lymph node with latent Propionibacterium acnes cells inside of them. Okay, now what can happen from time to time is that these latent Propionibacterium acnes cells, they can reactivate. So it's not very well understood, even as far as this theory goes, it's not very well explained, but something happens. The Propionibacterium acnes, maybe it senses that the macrophage is getting old and is weak, and it returns to the active state. So what can happen is it can reactivate then inside some of these macrophages now, and then once it's reactivated, its division rate will go back up. So what can then happen inside this macrophage is that the phagosome can end up full of Mycobacterium sorry, I keep wanting to say Mycobacterium tuberculosis. I've just said that bacterial name so many times in this video that now I'm just used to saying it. So the phagosome can end up full of the Propionibacterium acnes cells, like so. So here, this picture is supposed to represent that. So we've now got our macrophage, again coloured in yellow, now with lots of Propionibacterium acnes cells in the phagosome. Okay, so reactivation can occur, and then it's not really understood, it's not really explained in the theory what happens after that, but maybe the Propionibacterium acnes cells can actually burst the phagocyte open and then spread to other macrophages. Now, why is this called an endogenous infection and not just an infection? The idea is that this infection is not particularly dangerous at all, and that the body acknowledges normally that it is not very dangerous at all, and therefore doesn't launch the advanced adaptive immune responses against it. So you don't get the T helper 1 adaptive immune response initiated against the Propionibacterium acnes, and hence we refer to this as an endogenous infection, because it's presumed to be reasonably harmless and just something that's occurring there as though endogenous to the body. So the body isn't going to actually launch a horrendous adaptive immune response to the Propionibacterium acnes. So the idea is then that in normal people you have this endogenous infection of Propionibacterium acnes where the Propionibacterium acnes cells get phagocytosed by alveolar macrophages. However, some of them don't actually get killed because they can go into a latent state. Then they can wait a long time, and maybe the macrophage that they're inside of gets weak, and then they can return to the active state and divide inside the phagosome, potentially break the phagocyte open, and then they'll be phagocytosed by other macrophages, and those macrophages, many of them might kill the Propionibacterium acnes cells, and some of the Propionibacterium acnes cells might actually go into the latent state and therefore survive. And the idea is that just the macrophages can usually control this, i.e. that the P. acnes population will be kept under control, it won't go out of control, and that the presence of the Propionibacterium acnes is not particularly dangerous, so we call it an endogenous infection. So I hope that's kind of clear what's meant by P. acnes being this endogenous infection, and I'll state again that this is all just a theory, this is not widely accepted fact. Okay, so the theory that Propionibacterium acnes is an endogenous infection is what we've just discussed, not hard fact. Now, the idea then is that in sarcoidosis, what happens is the genetically susceptible host launches a hypersensitive adaptive immune response against the Propionibacterium acnes endogenous infection. So the idea is that because of your genetics, you are actually going to launch an adaptive immune response against the Propionibacterium acnes, even though it is not actually that dangerous. So this is what we would describe as a hypersensitivity reaction, if indeed this is the correct theory of sarcoidosis. Okay, so because of your genetics then, you are going to initiate an adaptive immune response against the Propionibacterium acnes, which is unnecessary. It's not necessary, and therefore it's a hypersensitivity response over the sensitive response. We are launching it against something that doesn't really warrant it being launched against. Okay, so because of your genetics, you're going to launch a hypersensitive adaptive immune response, which will be a T helper 1 mediated adaptive immune response. And the idea is you then form granulomas around the macrophages that have got the Propionibacterium acnes reactivating. So the idea is that the granulomas form around these macrophages where the Propionibacterium acnes cells have reactivated. So in sarcoidosis you get granulomas forming around these macrophages with the Propionibacterium acnes in. Okay, so that then is the Propionibacterium acnes theory of sarcoidosis, and I'll just state it again. So the idea is that in most people in the population we have an endogenous infection of Propionibacterium acnes in the lung and mediastinal lymph nodes, and this is because when the alveolar macrophages engulf inhaled Propionibacterium acnes cells, some of those Propionibacterium acnes cells go into a latent state where they can resist being destroyed by the alveolar macrophages. Then what seems to happen is they can reactivate long later, so a long time after, they can reactivate inside the phagosome and then start dividing inside that phagosome, potentially bursting the macrophage open and spreading to other macrophages. But of course, most of those Propionibacterium acnes will then be destroyed by the next macrophage in the chain, and only the ones that then go into a latent state will survive. So the idea is that usually this population of Propionibacterium acnes is kept under control just with the alveolar macrophages and the macrophages in the mediastinal lymph nodes. However, in sarcoidosis, what we think, what this theory states happens then, is that you launch T helper 1 mediated adaptive immune responses against the Propionibacterium acnes, that is this endogenous infection. And this is a hypersensitivity reaction. You shouldn't launch the T helper 1 mediated adaptive immune response against this bacterium because it's not actually dangerous. And the idea then is that whenever a Propionibacterium acnes chooses to reactivate inside the macrophage, that will then trigger a granuloma to form around it. So what's the evidence then in favour of this theory? Why do some people believe this theory? Well, one of the great pieces of evidence is that if you take sarcoid granulomas and you try to culture bacterial cells from them, one of the bacterial cells that you can always get to actually grow from the sarcoid granuloma tissue is Propionibacterium acnes. So culturing is one of the key pieces of experimental evidence in favour of this. So I'll repeat that. If you try to culture bacterial cells from sarcoid granuloma tissue, and remember I've described to you what you'd do there, you'd basically just spread sarcoid granuloma tissue over a agar plate and see what bacterial cells grow from that. And Propionibacterium acnes can actually be found in this way. Okay. Also, certain experiments where you can actually try to find the Propionibacterium acnes in the sarcoid granuloma tissue. So for instance with immunohistochemistry, so antibodies targeted against certain antigens of Propionibacterium acnes can actually demonstrate the Propionibacterium acnes inside the granulomas of sarcoidosis. Okay, so you can actually find the bacterium in the granulomas of sarcoidosis, which is certainly better than can be said for Mycobacterium tuberculosis. Okay, a second major piece of evidence in favour of this is that you can actually find macrophages in normal healthy people without sarcoidosis which contain latent Propionibacterium acnes. So the endogenous infection does seem to be true. So you can find both macrophages that look like this, where you seem to have a latent Propionibacterium acnes cell inside of them, and you can find macrophages that look like this, where you seem to have lots of Propionibacterium acnes cells inside the phagosome, and they seem to have divided. You can find these in normal people, so people without sarcoidosis, normal controls we'll call them. Okay, you can also of course find them in people with sarcoidosis, and in people with sarcoidosis these macrophages, the ones with the multiple Propionibacterium acnes cells in the phagosome, they are surrounded generally by a granuloma. So that is the evidence in favour of the Propionibacterium acnes theory of sarcoidosis. Okay, so that's where I'm going to end this video. To summarise what we have learnt in this video, we have seen that sarcoidosis is a systemic granulomatous disease in which you get these granulomas forming in certain tissues of the body, in many different tissues of the body. They can form in almost any tissue of the body, but the main place that they form is in the lungs and mediastinal lymph nodes. Another common place is in the skin and also in the uvea of the eyes. Rarely, you can get them forming in the heart, but the consequences can be extremely severe. They can lead to potentially fatal arrhythmias. Okay, we've then discussed the pathogenesis of sarcoidosis, and we majorly understood this by looking at the pathogenesis of tuberculosis because they're both granulomatous diseases. So that explains to us the T helper 1 mediated adaptive immune response and the formation of granulomas, and gives us some insight into why granulomas form and what their purpose is. Okay, we then saw the two final theories, the Mycobacterial theory and the Propionibacterial theory of sarcoidosis. The Mycobacterial theory says that sarcoidosis is just part of the spectrum of outcomes of a primary tuberculosis infection. Okay, so in sarcoidosis, the idea is that when you get exposed to Mycobacterium tuberculosis for the first time, because of genetic deficits in your adaptive immune response to Mycobacterium tuberculosis, because of your genetic makeup, you can't actually fight the Mycobacterium tuberculosis well enough to just get a latent tuberculosis infection. It's not quite bad enough that you get the progressive pulmonary TB or the miliary tuberculosis, but it's not quite good enough that you get a latent TB infection. And instead what actually happens is you get granulomas forming in the lungs, but they don't manage to contain the Mycobacterium tuberculosis. It gets out and then disseminates to other sites in the body, and then we get granulomas forming at those other sites in the body. So that's the Mycobacterial theory of sarcoidosis. The Propionibacterial acnes theory of sarcoidosis says that actually Propionibacterium acnes is a endogenous infection. There is an endogenous infection present in the lungs and mediastinal lymph nodes by this bacterium, Propionibacterium acnes, and that in normal people this doesn't cause a problem because they don't launch an adaptive immune response against it. Instead, in people with sarcoidosis, you get a hypersensitive adaptive immune response of the T helper 1 type against the Propionibacterium acnes, and you get granulomas therefore forming. And the Propionibacterium acnes theory would then say potentially these macrophages carrying the latent Propionibacterium acnes can go to different sites of the body and wherever they go, and you get the reactivation of Propionibacterium acnes, you'll get one of the granulomas forming. So that's why granulomas can form elsewhere. And its answer to remission would be, well, maybe something happens to end this hypersensitive adaptive immune response, and then you go back to treating the endogenous infection of Propionibacterium acnes like someone without sarcoidosis, a healthy control, would respond to the Propionibacterium acnes. But overall, it is unclear at the present time which of these is correct. Both of them might be wrong, and there might be some totally different theory to explain sarcoidosis, or alternatively both of them might be right, and it might be the case that there isn't just one form of sarcoidosis, and this is a concept that actually quite a few people are now starting to suggest might be the truth, that actually sarcoidosis is not just one disease. Potentially we might be looking at the sarcoidoses. There might be loads of different forms of sarcoidosis where it's different agents causing the systemic granulomatous inflammation. Okay, so with that then we will end this video on sarcoidosis.